Martin. Yeah. So really a wonderful opportunity to come here, India far away. As you know, I'm from China, but I studied in United States almost 25 years. It's a wonderful journey. And I'm very glad I have opportunity to share with you about the sickle cell disease metabolomic and new insight and novel therapy. So why metabolomic? So as you know, our body contains the DNA, RNA, protein, and then eventually go to small metabolite. So metabolite is in the pyramid of life. As you can see from here, we have a total like 25,000 genes, which is people largely do the genomic study, and very complicated. I hope Vivian can talk out more clear about the genomics. But for me, I'm more focused on the metabolomic because there's a unique feature of metabolomic. This slide show you is the pyramid on the top of the life. And more importantly, and the metabolomic approach, our laboratory has been interested in this metabolomic approach since 2008. And for almost like 8 or 9 years, we have built a pipeline to study metabolomic. So what's a metabolomic profiling? Actually, accurately measure functional phenotype that are the net result of genomic, transcriptomic, and proteomic change. And more importantly, metabolites are closer to the phenotype and they respond much faster to external stimuli than any other omic. So the metabolome or metabolomic platform technology is really improving. So basically you can connect the metabolite, small, get rid of DNA on the protein and RNA, and you purify the small molecule, use different methodology. Then you couple it with the HPLC or tandem MS, try to separate this small metabolite. And this is a larger scale unbiased. Then you will connect the data, then generate heat map, and after heat map, you will interpret your signaling path or metabolomic pathway, how they change. Using this high throughput unbiased technology, you can benefit to determine cellular and the tissue metabolism, how they change under the disease state compared to normal state. You potentially can identify the disease biomarker and pathogenesis of the disease development. And you also may be very important, like Martin studied the drug discovery, and I think it's very beneficial. Sometimes you discover the drug, you don't know what's the target. So you will likely use metabolomic and try, hopefully, to identify mechanistic toxicology and the mechanism of action. So the overall goal for the metabolomic field, basically want to really try to interpret or determine how the signaling pathway and the eventual metabolic pathway. So as you can see, our body is so complicated, but all the metabolite is regulated by specific signaling pathway. So I would like to give you some example. My laboratory used this approach to study sickle cell disease. So my laboratory, since 2006 and almost 11 years, studies the translational study in sickle cell disease. And the goal, of course, is try to identify mechanistic target to treat and prevent sickle cell progression. We know this is a single gene mutation, but as you colleague here, the sickle cell, which can affect the multi-organ damage. So then can we slow down even they have this mutation and the disease progression and organ damage? So what's the strategy we took back to almost 2008 and more than 9 years ago is use metabolomic screening in sickle cell Berkeley mice coupled with the sickle cell human translational study. And this mice, as you heard, is a humanized mouse model. It's very unique and close to human sickle cell disease. So first the data we actually very fortunately and successfully detected using metabolomic screening. We identified the 251 metabolite in the blood of sickle cell mice. And I want to choose the two molecules which we started very interesting in the beginning. One is adenosine, which is known induced under hypoxia. Another one is ERISA-specific allosteric modulator 2,3-BPG. So when I got the new metabolite and detected in the by metabolomic, the normal way I taught myself, also taught the student, you should learn how it is produced, and then you need to learn what's the literature reported the function. So as you can see, the 2,3-BPG in the biochemistry textbook in the medical school, you may, or in the PhD biochemistry textbook, you learn is produced in the glycolysis pathway, especially in the urethral. There's a specific the rumporenubrin shunt, which produce the 2,3-DPG, then come back to 3-PPG and go back to the glycolysis. 25% of glucose used to generate this 2,3-PPG close to millimolar concentration as a hemoglobin. So this is how it's produced in our body. The major function is 2,3-PPG is endogenous allosteric modulator, promote oxygen release, binding the tetramer of the hemoglobin, and once the 2,3 binds to the central water cavity, cause conformation change, induce oxygen release by decreased hemoglobin oxygen binding affinity. How about adenosine? So it's the same thing. We need to learn how it's produced, what is the function. As I mentioned, the adenosine usually induced under hypox condition by two actonucleotides from ATP, from the usually ATP stored inside the our cellular inside of the cells. But under the hypoxic stress condition, ATP can release from intracellular to extracellular, then convert to adenosine by two actonucleotides on the surface of the cell surface. And the adenosine is not only just a simple small nucleoside, it functions as a single molecule, comes through the full adenosine receptor, which is a GI or GS coupling receptor. Is known in the textbook or in the literature. Activation of A2B can cause vasodilation, anti-inflammation, and anti-vascular leakage at the acute stage. However, the biological function of increased adenosine in the red blood cell, even sickle cell disease, remain unclear. So we published a paper around 2011 by the assistant professor, Dr. Yunjin Zhang, in my laboratory as a leading author, and demonstrated in the sickle cell condition, and increase the sickle due to the mutation caused the hemolysis. Hemolysis induced ATP release and converted to adenosine. Adenosine synchronous to A2B receptor, which is a GS coupling receptor, induce pKa. pK activation induce 2,3-DPG, also 2,3-BPG production, and this increase oxygen release due to the decreased hemoglobin oxygen binding affinity. This will increase the deoxypolymerization of the sickle hemoglobin, and this will form malicious cycle, and then cause the disease progression. So we can interfere this pathway either by lower the adenosine by PEG-ADA, which is FDA-approved drug, to lower the adenosine treat ADA deficient human. We also can specifically interfere A2B receptor by A2B antagonist to block this malicious cycle and block the disease progression. Another example I want to share with you, using this unbiased metabolomic screening, we also identified, you can see the lipid substantial shift. This red dot is a sickle cell, and the blue is a wild type of mice. And one of the major lipid highly induced is sphingosine 1-phosphate. So I mentioned the one we got the new molecule, the methodology for us to learn is how it's produced, what's the function. So in the literature, in the red blood cell, very unique. So to test that, we basically conducted the mouse work and the human and the translational study, demonstrated hypoxia can induce ACPHK1 activity very fast. Then this can increase ACE1P, and the ACE1P induction can induce sickling and form malicious cycle very similar like adenosine. So, but the remaining question is how ACE1P and induced sickling still unknown. I will talk about some unpublished data and in our laboratory. And moreover, and the talented student in my laboratory also further demonstrate adenosine can synchronize A2B can through the pKa pathway phosphate ACPHK1 result the activation of ACPH normal and the sickle red blood cell. So the major question remaining is what's the mechanism underlying detrimental effect of ACPHK1 in sickle cell disease? So we demonstrate increased ACE1P due to activation of ACPHK1 contribute to sickling, but the potential mechanism remain unknown, which is also possible to the whole field, how and why ACE1P induces sickling. So in order to demonstrate in order to answer this question, Kai-Chi spent 2 years to generate this double deficient mice. Basically, by mating sickle cell with the ACPHK1 global deficient mice, and we validated the genotype, and also we validated the ACPHK1 is gone in the sickle red blood cell, and the ACE1P also reduced, and the plasma ACE1P, and they almost decreased, compared to the induction of a sickle cell and closed white. Because there's an ACPHK2 also can produce ACE1P. And then we go back to look at the sickling, and the deletion of ACPHK1 and the substantially reduce the sickle and the reduce the hemolysis, prolong the lifespan. And also the splenomegaly also substantially reduced, multiple tissue damage also reduced. More importantly, and as I mentioned, the 2,3-BPG and is known induced in the sickle condition and likely contributed to the sickling. And we found that the sickle reduced, hemolysis reduced. So we go ahead and measure the 2,3-BPG is also reduced in the double deficient mice by getting rid of ACPHK1. P50, which is oxygen hemoglobin binding affinity, also significantly reduced. And the reactive oxygen species also substantially reduced. So the question remaining, how does elevate ACPHK1 lead to 2,3-DPG induction and reduce reactive oxygen species? So to address this question, we actually took the unbiased approach to conduct metabolomic screening again. Basically, we compare white type sickle cell double deficient mice and the erythrocyte metabolomic profiling. We demonstrated the ACPHK1 deletion correct the pathogenic metabolic reprogramming by promoting pentophosphate pathway and reducing glycolysis. So in order to have a better understanding, I would like to share with you about basically, and when you look at the detailed metabolomic screening, you really can tell each metabolite, how they change, how they altered, and you can interpret which potential enzyme may be interfered or altered. So you can see here our metabolomic screening really tell glucose, which is the only metabolite can use by red blood cell because the red blood cell has no mitochondria. So glucose as only sources metabolize to the glycolysis or to the pentophosphate. And by using the metabolomic screening, we demonstrate the ACPHK1 deletion lead to metabolic bottleneck at the G3P, glycerol 3-phosphate stage, and this actually glycated step and switch glucose flux because this bottleneck, and so this go back and then shunted to pentophosphate pathway. So because of deletion of these, and we actually basically correct, increase the 2,3-BG in the sickle cell, and also partially enhance the pentophosphate pathway, and the pentophosphate is a very important pathway to generate antioxidant. So this actually, and because it switched to pentophosphate pathway, and this reduce oxidative stress, potentially also reduce glycolysis, particularly in this pathway, and the result decrease the sickling by decrease the oxygen release and to the peripheral tissue. So this also potentially indicated because is this pathway blocked, the key enzyme involved to convert G3P to 2,3-PG is GABA-DH, which is a very important enzyme in the glycolytic pathway. And this also implicates that GABA-DH should substantially reduced. So actually in the erythrocyte field, as you heard from Mohan's talk, the membrane anchored protein is extremely important. Recent years, for 10 years, erythrocyte metabolomic membrane structure biology has evolved substantially, particularly and in the erythrocyte. Erythrocyte is very unique, contains the oxygen, carry the oxygen, but never use it. Therefore, glucose and either go to pentophosphate, generate antioxidant, or shunted glycolysis is finely regulated. In the low MOX condition, and the GABA-DH, which is the key enzyme and involved the glycolysis, is 70% the GABA-DH actually anchor the membrane to the band 3. However, in the hypox condition, and this GABA-DH actually can release from the membrane go to the cytosol by the deoxyhemoglobin and binding to the band 3. By this switching and the deoxyhemoglobin binding with band 3, lead the GABA-DH release the cytosol and increase the glycolysis and decrease the pentophosphate pathway and allow the more oxygen release. So in the literature, so we do know erythrocyte metabolism is regulated by oxygen availability, and also the red blood cell glucose metabolism is programmed by GABA-DH localization. Because initially in the lomoxia, membrane-anchored GABA-DH is not available for the glycolysis. However, under lomoxia, because the increased oxygen release, deoxyhemoglobin anchored to the membrane and allow the GABA-DH release. However, the fine regulation remains unclear. What specific molecule involves this process? And I do not have time to talk about too much detail, but we do identify that sphingosine 1-phosphate is a major rate-limiting step to control this trafficking of the GABA-DH from membrane-anchored to the cytosol, result in increased glycolysis and increase the glucose shunted glycolysis. So ACE1P is essential for deoxyhemoglobin, Sickle the anchoring to the membrane and releasing of GABA-DH from the membrane to the cytosol. More importantly, and by collaboration with Martin, we have a very successful collaboration, and he has demonstrated ACE1P bound with the 2,3-BPG hemoglobin A to cause a further conformation change and the stabilization of deoxyhemoglobin. So this is a crystal structure, and he has done with his talent poster in his laboratory. So I want to just use this model to summarize our current work. Under Sickle condition, due to the hypoxia, deoxygenation, and the Sickle hemolysis, ATP released, and the released ATP will convert by the CD73 to the adenosine. Adenosine signal A to B and lead to the phosphorylation of the pKa because it's GS-couplin receptor, and this will likely induce the phosphorylation by the BPG mutase, one of the key enzymes, and allow the 2,3-DPG induction, and this will induce the deoxygenated polyhemoglobin and cause the sickling and the reactive oxygen species. And this will actually lead the deoxyhemoglobin become more stabilized as a deoxygenated state. Meanwhile, we identified the A to B singlet pKa and allow the ACPHK1 activated. And this will generate the ACE1P. ACE1P actually as an anchoring can allow the deoxyhemoglobin and stabilize as a deoxy form, and this allows the GABA-DH anchored membrane release, and this will induce metabolic reprogramming by enhanced glycolysis, decrease pentophosphate, and this will increase more sickling and more reactive oxygen species production. So finally, I want to just comment on this work actually point out in the long erythroid condition, adenosine and ACE1P likely as a hypoxic sensor because it quickly induced under hypoxic condition and implicated in such as physiological and pathological hypoxia that affect almost every organ in the human body. So by discovering this in the sickle cell, we extended by involved a very nice, elegant work and LT-omics study had by the UC Denver, and 25 scientists from 6 countries involved. Basically, and we bring the normal individual to the South Bolivia from sea level, 21 healthy individual, half male and half female. And basically, we after the different time points, we connect the red blood cell from human at the sea level, high altitude. We did the metabolomic screening. We measured the red blood cell oxygen release capacity. And we actually, for the time, I would not talk about the detail, but we recently published 3 papers and demonstrated the pathway we observing in the sickle cell condition also exists in the normal individual, such as under high altitude condition. However, because this pathway is beneficial in contrast to sickle cell, because this normal individual, there's no mutation. So therefore, this pathway can be potential and therapeutic possibility. So in conclusion, We have demonstrated using the unbiased screening and coupled with the genetic mouse study and human translational study, we demonstrated adenosine signaling with A2B coupled with PKA and ACPHK1 are detrimental in sickle cell disease to induce metabolomic reprogramming, sickling, and reactive oxygen species production. Inhibition of this pathway are effective therapy potentially for sickle cell disease, and we currently collaborate work with LPAS to treat with a neutralized S1P antibody. And we also demonstrate that this pathway can be beneficial for normal individual under hypoxic condition, and this enhance this pathway can be potentially important therapeutic possibility. Finally, I want to thank all the trainees and the postdocs involved in this work, particularly Atal and sitting here, and he was trained in my lab for 3 years, and then come back to the India, now in one of the best top largest school and the university at Hyderabad. So I'm really happy to see you here. And also this is my lab and in the past and the current, and also funding. Thank you.